Hello, everyone. I'm Sterling Wright. I'm a PhD student at the Penn State University. And in this presentation, I'll be going over some of the work that I've been doing at Histria, which is, as you can tell in my title, a Greco-Roman site along the Danubian Frontier. I also just want to take the time right now to also thank the organizers for putting all of this virtual meeting together. And I know it takes a lot of effort, and just want to make sure that they know that I appreciate that. All right, let's get started. So here's a map of where Histria is located. As you can see here, it's situated near the mouth of the Danube and the western edge of the Black Sea. And just for reference, Histria is about 60 kilometers away from Constanza, which was known as Tomis during the ancient Roman period. And you may know that the poet Ovid was actually exiled there. And like, if you were to tell, like, just read from his accounts of Histria or of Tomis and the area, you may not have like a great view of it, but I can tell you from my experience there, it's been actually quite pleasant. So Histria was established by ancient Greek colonists from the city of Miletus in Asia Minor during the 7th century BCE. And during the Roman period, we have literary sources and archaeological evidence even to suggest that populations from the Balkans, the steppe, Anatolia, and other places in Europe will actually come here to trade and like occupy the site and do all sorts of stuff, including actually like pillage and raided even. So, however, because of these raids, the site will actually be would have been abandoned by the 6th century CE, and it would never have been reoccupied since then. The city was mostly forgotten until the Romanian historian archaeologist Vasile Parvan started his excavations there in 1914. His projects there focused mainly on this area, the Acropolis, and located right behind the late Roman walls. But later excavations that would take place between the 1950s and the 1980s would turn their attention to the plateau. And this is the area that I've been working in. And this area was a walled residential area in the early phases of the settlement, but would actually be turned into a graveyard during the Roman period. And so just to warn you, I do have human remains on the next slide. So like many classical archaeological excavations, human remains at the site were mostly ignored and like poorly documented because throughout the 1900s, archaeologists were mostly interested just in the material goods. However, over the past few decades, there has been a renewed interest in these remains. For instance, Kretsu et al. In 2020 conducted a bioarchaeological analysis on burials dating to the Roman period to gain new information about the possible relationships between the individuals. They perform anthropological analyses such as estimating the age and the sex of these remains, as well as investigating the funerary structures and the grave goods associated with the individuals. The analyses suggest that the burial traditions at Histria were quite diverse during the Roman period, and potentially their findings indicate that Histria could be thought of as a multi-ethnic population. These types of findings and research methods have been instrumental in my line of work. So with the rich information already associated with these burials, I'm hoping that my methods and my analyses could offer additional information to give us new insights about the lives of these past individuals that were buried at the site. In essence, I'm thinking about like how could applying biomolecular methods could help us further understand whether like certain cultural groups are at higher risk for social oral diseases than others. Could they also provide us new information about how the social identity of individuals intersect with the genetic identity? And then I'm also interested in understanding like how could these methods also help us understand how the biological sex, according to the XX and XY chromosomes, play a role in understanding the population demographics or dynamics at Histria. So for this project that I've been working on, I was interested in recovering biomolecules from the dentin, the dental calculus, and the enamel. Each of these parts of a tooth, a single tooth, can provide specific information. For instance, human DNA can be recovered from the dentin, which can be used to recover genetic ancestry information. The enamel can be used to recover sex-specific amyloidogen peptides in order for us to determine sex. And then dental calculus, which is my main area of focus, includes both microbial and host DNA as well as food particles, but also it can give us information about how cultural transformations or transitions occurred over time. It can also give us new insights about ancient migrations, as well as insights into people's health. So, however, while a great deal of information can be gained by applying these different biomolecular techniques to the different parts of the tooth, there is no guarantee that you can gain results by applying them, as all of this sort of work is dependent on preservation. Many factors such as climate, the annual mean temperature, the temperature fluctuation patterns in an area, in the geographical area, the age of the samples, and how they were treated after excavation can all influence whether or not the biomolecular preservation is robust. In general, places that are hot and humid are not really well suited for biomolecular preservation, and therefore scientists often struggle with recovering ancient DNA as well as paleoproteins from these areas, and that's what this map is really showing here, as where each dot represents a place where scientists were able to recover ancient DNA from remains, and you can see there's much of the concentration of many of the samples are from more temperate climates. And so while this figure is outdated and the methods in ancient DNA are continually being advanced, and scientists are being able to have more success to recover ancient DNA from places like Mesoamerica and across the African continent, preservation is still critical to assess before starting or initiating larger scale programs. And so with that being said, I found it was very important to study the biomolecular preservation at Histria before pursuing a larger scale project where I would take, like hundreds of samples and just applying them, these biomolecular technologies to them without having any idea whether or not the biomolecular preservation is actually robust or not. And so by just establishing a baseline on the preservation on a small number of samples, I was hoping that this gives me some idea of whether or not the preservation is adequate. And then if the results are promising and robust, then I could apply these techniques onto a larger sample size and then be able to address much larger and broader anthropological questions. So this project of mine started in 2018 when I participated in archaeological excavations at Histria, working with Dr. Edward Benowitz at the University of Texas. He's really the archaeological director at the site, and he's organized this like multidisciplinary project where he's bringing in physical anthropologists, ancient DNA scientists like myself, radiocarbon dating, chemical isotopic researchers to really like address these much broader scale questions at the site by looking at the human remains that date to the Roman period. So for my part of the project, I was working with him. I was also working with Andrei Soficaru, because as I mentioned earlier, people have been excavating at the site for almost like 100 years, and the human remains have been like recovered like 50, 60 years ago, have been housed at the institute here in Bucharest. And it's there where I was working with him and identifying what potential dental calculus and tooth samples could be used for ancient DNA and paleoproteomic analysis. So we identified 5 calculus and 5 dentin samples, which were then shipped to the University of Oklahoma, where I was at the time conducting my master's thesis. So once the samples were shipped there, they were obtained by our facility, the Laboratories of Molecular Anthropology and Microbiome Research, where we only do ancient DNA research there. And although I had 10 samples, unfortunately there was some equipment failure during the DNA extraction process that failed, and so I had to redo the whole process. But due to budget constraints, I was only able to do it to 2 dentin and 2 calculus samples. And prepared the DNA extracts. I did some library preparation and then some DNA sequencing so that I could do this downstreaming processing, including data quality assessment and filtering, damage pattern assessment, microbial profiles reconstruction, mitochondrial haplotype assignment, and sex estimation. I was also able to apply the paleoproteomic analysis, which I'll get into a little more detail here in a bit, 3 samples, including 1 juvenile sample. So here are my basic research questions for this project. So I wanted to know, like, do the dental calculus samples have oral microbes? Is the DNA in the dental calculus and dentin actually ancient? Is the mitochondrial DNA from these dentin samples preserved? And then can we estimate the sex of individuals with a proteomic analysis that's actually minimally destructive? So first analysis I ran on these samples was horse tracker. And this is just a Bayesian tool that basically we use in ancient DNA to assess whether or not the microbial DNA in our calculus samples are oral by nature or not. And so I applied these to not just my calculus samples, which are here labeled with C's on the ends, but also my dentin samples, which here on the D on the end. I was just curious to include them here just to see what kind of results I got. And so what these results tell me is that the microbial DNA in my 2 calculus samples are mostly oral, and they could be tied to calculus, which was really promising and reassuring to indicate if I wanted to later down the road, like perform like more high-level analyses that these samples have the right type of microbial DNA to do that analysis. It was a surprising finding here that one of the dentin samples actually had a pretty significant proportion of calculus in them. And one explanation that we think that could explain this was that it was probably just invisible traces of calculus on the tooth that we just didn't see when we actually did the DNA extraction. And like this result here for this dentin sample was expected, and like because you're not expecting to find oral microbes on the tooth itself. These results were they're promising because of we also included an extraction blank here on the end, and because there was no oral DNA identifying it, that suggested to us that we didn't find any cross-contamination in that respect. So source tracking gave me a broad overview of what oral species, what of the overall communities, but didn't tell me what exact species were in them. And so what I have here is a list of species that are identified in my 2 calculus samples. And all these species can be found in the human oral microbiome database, which is very promising, suggesting that, again, that the calculus samples have like very robust preservation. What's also interesting to discover here is that some of the species are associated with periodontal disease. So Danubian Frontier and Treponema denticola have been associated with the red complex. And what that entails is that these species have been highly associated with people with periodontal disease and gum disease. Another species that was pretty interesting to find was Analysia oral taxon 439, because this species has been also identified in other ancient DNA datasets and has like a strong temporal and geographical signal. And so this like phylogenetic comparisons of this bacterium by archaeological populations in Italy, the Balkans, and Japan have shown that this bacterium is not just, it's tied like to very specific cultures. So it can be tied to like hunter-gatherers, to agriculturalists, both in Italy and Balkans, as well as in Japan. So with that being said, I was able to like do a positive check mark for my first question. Says because it looks like there are oral microbes in my dense calculus samples. But at this point, I didn't know whether or not they were ancient. And so my next step was to, my next question was like, is the DNA in the dense calculus and as well as my dentin samples are ancient? So I use map damage, and this is a widely used tool in the ancient DNA field to address whether or not the DNA in your samples are ancient. And so I won't go into a whole lot of detail here, but the main takeaway here is that ancient DNA has a very different characteristics than modern DNA. Oops, sorry. Here we go. So if you have ancient DNA, your plots here should have what we call usually a smiley face, whereas modern DNA usually has just a flat line. And so when I was looking at my dental calculus samples, when I mapped them to like microbial references as well as to the human references, they have these types of smiley plot shapes to them. Whereas when I map my dentin samples to a human source, they also have this sort of a smiley plot characteristic. But when I map the human sources to the dentin samples to microbial sources, they have these more flat line, and that's what is expected because there's not that much microbial DNA in the dentin samples. And now it's not. All right, here we go. And so based on those results, I could say that the DNA in both the calculus and the dentin samples is ancient, which brings me to my next question was whether or not the mitochondrial DNA from the dentin samples were preserved. So mitochondrial DNA is really important because it helps us or allows us to trace the matrilineal ancestry of individuals as well as their belonging respective populations. And so I was able to identify both haplotypes for both dentin samples, and both of these haplotypes have been identified in other ancient DNA datasets. The second dentin sample I have here, gosh, it's not working. Was actually identified. This haplotype has also been identified in the high frequency in Iron Age Scythian populations. So because I'm doing with the Roman period here, I can't say whether or not that this individual had Scythian ancestry or not, but I can say that it has the same haplotype that was also in a high frequency found in other Iron Age Scythian populations. All this is also like confirmed with the map damage results again that the mitochondrial DNA is also ancient. And so I was able to checkmark my first three questions, and then so this brings me to my last question. It's like, can we estimate the sex of ancient individuals with a proteomic approach? So for this part of the project, I was working with Christian Rayfield, who was a PhD student at Oklahoma University at the time. And what we were really interested in is using like this acid solution where we would dip the tip of the enamel into the solution. And so unlike other biomolecular techniques where it's pretty destructive, and you're just like, for most cases, sawing off or removing completely and destroying the biomaterial. Here, we're just dipping it in a solution, the enamel in a solution to recover the peptides. And so most of the tooth is still intact. And we're interested in recovering specifically MLX and MLY peptides because these peptides are encoded by the amylogen X and amylogen Y genes. And so if we're able to recover like only AML-X proteins or peptides from the enamel, that would suggest that the individual is female, whereas if you were able to recover both MLX and MLY peptides, then this could suggest that the individual was a male. Now, with that being said, just like everything else with ancient DNA and other sorts of work, there is a case of dropout with this. So just because it's the absence of MLY doesn't necessarily that it wasn't necessarily just dropped out. There's a false negative rate potentially, but this is like proteomics. These methods are quite been tested on and have been quite robust in other studies. And so what we're able to identify that two of our all three individuals that we tested this, and I don't have the results here, but also, like I said earlier, we also applied this to a juvenile. Had the presence of MLX peptide, whereas only one individual had MLY, and our other individual here, as well as our juvenile individual, did not have this MLY peptide.
Thank you so much, Sterling. We're gonna have to wrap it up, but if you have
Okay.
more information, you can drop it in the chat.
Okay.
And we'll also have time for
Sorry, yeah, I think
questions.
the clicker on here is it's throwing me
That's
off.
totally fine. If
Okay. So yep, thank you. I just want to thank all my collaborators and everything, and thank you.